Now we're back at the NMJ, but this time we'll introduce a pipette through which we can inject a calcium-rich solution into the area right around the presynaptic terminal. This kind of flow is fairly common for an in vitro, or out-of-the-body preparation, since it allows oxygen and nutrients, and whatever else is needed to keep the prep alive, circulating. However, a happy consequence of this kind of arrangement is that it's easy to control both the concentration and timing of the release of a solution through our injector pipette. Basically, we can puff our calcium solution, or any other solution for that matter, into a particular spot in the prep at a particular time, and because of the flow in the chamber that tends to wash it away, the effects will be relatively confined in both space and time. And since one can construct the bathing solution to contain any desired ionic makeup, it's possible to deplete the bath solution of our ion of interest, so that the puff pretty much completely controls the concentration of that one ion. In testing a variety of different ions, cats found that there was a surprising and precise dependence between EPP amplitude and the concentration and timing of calcium in the NMJ. Recall that when the presynaptic fiber is stimulated, a full-sized end plate potential, many times larger than the mini EPP, is evoked. Katz and his colleagues found that when they puffed different concentrations of calcium around the NMJ just before stimulating the presynaptic nerve fiber and recording from the postsynaptic cell, the size of the resulting EPP depended strongly on the precise concentration of calcium present at that time. In particular, the amplitude of the end plate potential was approximately proportional to the calcium's concentration to the fourth power. This suggested that calcium plays some role in determining how big a postsynaptic potential is. Katz and company also noticed that it mattered when the calcium was puffed. If the calcium came after the presynaptic nerve was stimulated, no EPP was observed. So calcium not only determined how big the EPP would be, but it also needed to be present extracellularly when the presynaptic action potential arrived. Otherwise, it would have no effect. This is consistent with the idea that voltage-gated channels opened by the presynaptic depolarization allowed calcium to flow in, and that the then available intracellular calcium somehow allowed the release of neurotransmitter. To test our understanding of calcium's role, let's consider an experiment where instead of puffing calcium outside of the cell, let's imagine we have a pipette that allows us to inject calcium directly into the intracellular space of the presynaptic terminal.